So today I get to introduce Dr. Michael Bradshaw. He is here in the Department of Entomology and Plant Pathology. So Michael got his bachelor's degree at the University of Delaware, and then moved to the University of Washington in Seattle to do a master's degree and a PhD, master's degrees in plant sciences and a PhD in plant pathology. He then moved on to do 2 postdocs. So he did a shorter postdoc at USDA and then moved to Harvard and did a postdoc in mycology at Harvard. He came to us at NC State in 2023 as part of the cluster hire program into the Emerging Plant Disease and Global Food Security cluster. He joined, as I mentioned, the Department of Entomology and Plant Pathology. He also is the director of the Larry F. Brand Mycology Herbarium here at NC State University. And I think he's going to tell us a little bit about that as part of his work.
Yeah, thank you. Thank you. I appreciate it. So the title of my talk today is Using Herbarium Specimens to Uncover Fungal Diversity, Disease Spread, and Potential Extinction Events. And thank you guys for having me. You know, I always love talking about my research. My research centers around this fungal pathogen right here. This is powdery mildew. This is under the microscope. And I just think it's such a beautiful organism. I really think it's got to be one of the most beautiful organisms around. And some evidence for that is that this picture in the middle was taken by one of my students, Christian Shaw. He's not here. He submitted this. Do you guys know the NC State photography competition? So he submitted this one, and then one of my staff members submitted theirs, and my staff member got first place, and Christian got an honorable mention. But I want to just point out that they used his picture to showcase the webpage. So even though he got the honorable mention, I think that counts as some bonus points. And you guys can feel free to ask questions during my presentation.
Sorry.
Okay, so just an overview. I'm going to start off with an introduction to powdery mildew. I recognize a couple of you guys from my talk before, so you've probably heard this already, but hopefully powdery mildew will be ingrained in your head as well as its life cycle. Then I'm going to talk about powdery mildew biodiversity and why this is important and how I use herbarium specimens to better understand its diversity. Then I'm going to go into 2 systems that I've been studying heavily, and that's powdery mildews on oaks and powdery mildew on blueberries. Then I'm going to talk about a little study I did during the pandemic that I just think is really interesting, and it has a big genomic aspect, so I felt like you guys might be interested in it as well. And then I'm going to talk about a couple potential extinction events that I'm really trying to understand, because I just think that is, to use herbaria to understand extinction events is a really awesome use of it. You know, in this time, I don't know if you guys have heard about what happened with Duke, but they are getting rid of their herbarium, and there's a lot of noise about the importance of herbaria, and I think it's really important to show how herbaria can be used for more than just these classical taxonomy and morphology papers. And so that's one of my main research goals. Okay, so what exactly is powdery mildew? It's an obligate fungal pathogen it's monophyletic within a single family, Ehrzyphaceae. An obligate pathogen means that it needs a living host to survive. And that's probably why it's extremely understudied. You can't culture it. It's never been done before. There's probably just too much crosstalk with the plant and with the pathogen. It's literally everywhere. It infects over 10,000 species of plants. This is just angiosperm plants. If you guys walked outside this building right now and you looked for it, you'd probably find 20 different species just within a 50-yard radius. Yeah, it's one of the most prevalent plant pathogens in the world. Once you notice it, you can't unsee it. I always say that I'm trying to cure powdery mildew blindness. Has anyone heard about plant blindness? It's the inability to recognize plants in your environment. You know, you just see green because you don't understand all the different species. So you guys probably don't even notice the powdery mildew, but after today, I'm sure that it'll start showing up in your minds. And it's really common in North America. It really likes our temperate weather, and over 300 out of the estimated 906 species have been reported in North America. So why did I choose powdery mildew? So first off, just as I mentioned, it's really understudied, and I think it's so interesting, the coevolution that's going on between the powdery mildews and the hosts. And just some evidence about how understudied it is that this is a powdery mildew that I found during my PhD. You can see the plant is completely covered. It's this white all over the plant. This was found right outside the biology department at University of Washington, where there was a mycologist. He had never collected it, and it turns out this was an undescribed species. And something else I just want to mention, because you were talking about GenBank. And so there's a genus of powdery mildew, Phylactinia, and we're redoing this whole genus, and we submitted some of the first sequences to GenBank of Phylactinia. And they rejected all of our sequences. They said this is too abnormal. They said it was pretty much like alien DNA. I think the closest match was like 70% with a lot of other organisms. But we told them, this is the group that we study. The phylogeny matches the morphology, and they accepted our sequences. But just to give you an idea how understudied this group is. And I just love this quote too. This is from a really well-known mycologist, Amy Rossman. And she wrote that for some types of organisms, such as microfungi, New York State's forests are almost as unexplored as the tropical forests. And I always like, you can take this one step further. And if I went and collected specimens all around North Carolina State University, I would probably find between 15 and 20 undescribed species. And you could say that North Carolina State is as unexplored as the tropical forest. And then here's the powdery mildew life cycle. We have this asexual stage in the upper right corner. It consists of these conidiophores, which is a chain of spores. Each spore can travel around 200 miles. And you can have tens of millions of spores on a single leaf. And then we have the sexual stage. So most fungi have an asexual and sexual stage. And this consists of the fruiting body known as the chasmothecea here. And within it, you get the ascus, which holds the ascospores, which are the product of meiosis. One of powdery mildew's claims to fame is that, in the 1800s, they used to think that this was two different organisms. So they would give a name to the asexual stage, and then they'd give a name to the sexual organism. But in this great piece of work in the 1850s by the Chulosny brothers, they showed that this was actually the same organism, and they used powdery mildew as one of the main fungi to prove this. And why it was so easy is because powdery mildew switches to this sexual state when the weather starts to cool. So this is their resistant structure and allows them to survive the winter. So in the middle of, usually in the fall, they'll switch from this to this in the same spot in the same leaf. It's very obvious that this is the same organism. And I love to show this slide because it just shows that nobody is safe in the mycology world. So the fungus connects to the plant, and of course there's going to be a fungus that connects to the fungus. So this is one of those conidiophores that I showed you, and this is another fungal pathogen that attacks powdery mildew, and this is called Empelomyces. And the reason I'm showing you this is because this is something that really interests me. If powdery mildew is understudied, this is even more understudied. Here's another picture. And I mean, just imagine the comparative transcriptomics you can do of the plant, the pathogen, and the pathogen of the pathogen. And so I think that this is something that I'm trying to dive into more. There's only 2 cultures of this that have been isolated in North America, even though you find a ton of diversity in their genomic sequences. Okay, and then another thing I just want to mention and go over is because, you guys are at a university, you probably understand this, but I like to really emphasize why understanding powdery mildew biodiversity and taxonomy is important. And I just have a little case study that emphasizes this point. So I just want to talk about Ehrzyphi piscii. This is a powdery mildew that's host-specific to plants in the Fabaceae. This is mostly pea plants. And in the past, they used to think that this affected many different powdery mildews, but we've shown now that it's extremely host-specific. This is Ehrzyphi necator, and this is host-specific to grapes. You can't get it to infect any other powdery mildews. So I had a company that came to me, to ask me to do these fungicide efficacy trials, and they said they haven't been able to do this with powdery mildew, and they want some help. And they said, it's on the label, so that's the law, but I need to be able to control Ehrzyphi piscii to control on grapes. And if you remember, Ehrzyphi piscii only affects plants and legumes. So they clearly didn't have the taxonomic knowledge that was needed to run this experiment. So I told them that's impossible. I can't run this experiment for you. But I told them what I could do, and I explained it to them, and they understood that that's why they haven't been having success. They were going out in the field and collecting powdery mildew in all these different plants and trying to inoculate their grapes. So then they hired me. It's also really important for early detection. There's a lot of research that's going on now where people are putting up spore traps and trying to detect the pathogen before you see symptoms or signs of disease. And this way that you can spray your fungicide before it's damaged. And you need really specific primers to do this. And you can't do that if you don't have an understanding of all the different species. Same with fungicide timing. There's some organisms that are resistant to fungicide. Some powdery mildews come out at different times. So it's really important to understand all this different species. Also, how it's spreading across the world. And this is something that I like to do with my research. So I discussed a little bit about how powdery mildew was everywhere. But now I just want to show you some data proving this. So there's this big database through the National Fungus Herbarium in Washington, D.C. And within this herbarium, they keep a database of all the different fungi that have ever been reported. And within their database, they found that there were 72,336 powdery mildew reports. And this was 8.7% of all the reports in the database. So they have around 1 million fungi in their database, and almost 10% of them are powdery mildews. So 1 out of 10 of every fungal specimen that is collected and that has been reported is a powdery mildew. And around 2.5% of all angiosperms are estimated to have been reported as a powdery mildew host. We just see it everywhere. I'm growing a plant in the growth chamber in my lab right now to try and grow a rust, and we are finding powdery mildew infected on it. So we can't escape it. And then this is just a figure that we made from this database showing its geographic spread. All I'm trying to really point out here is that each different color is a different genus of powdery mildew. So the blue is all Ehrzyphi. It's the genus that contains around 50% of all powdery mildews. But the real take-home message here is look how small the circles are. This is the sample size in South America, Africa, Middle East, and Australia. And this is just showing that these are gaps in our knowledge. This is something I'd really like to study further. And then here, this is just powdery mildew hosts by genus. Each different color is a powdery mildew, is a host family, so is a plant family. And what you'll notice is that some genera only have one color, so they're host specific to a single family of plants. And this is really common with powdery mildew. As molecular techniques are being refined and the more sequencing we're doing, the more host specific we're finding these organisms are. And now I want to talk about my work in North American herbaria. So herbaria hold a wealth of undiscovered fungal diversity. I think that they're such amazing resources. Especially nowadays when if you want to get into a good journal, you really, you guys have probably all experienced this, that they really want you to kind of take a worldwide approach. And I realized with my PhD that it was pretty easy to take a worldwide approach when you have these herbaria specimens available to you. I could not leave my office now and tell you about specimens all throughout the world and look at their genetics. So we're getting better and better at sequencing. So just within North American herbaria, Mycoportal is our big database. We found that there's 97,506 powdery mildews. So these are collected all throughout North America and all throughout the world. And this is the herbarium here. This is the herbarium. I'm a director of the Larry F. Grand Mycological Herbarium. It's in Gardner Hall, right directly across from here. It contains around 8,000 specimens, and of those 8,400 were powdery mildews. We sequenced all of them and found 6 undescribed species, which we're trying to, we have a paper that's just about to come out now. Most of these were collected from North Carolina, but I'm also trying to really expand this collection. So there were 2 professors, Dr. Uwe Braun and Dr. Suzumu Takamatsu. These are kind of like the powdery mildew legends. I'm kind of following in their footsteps. And they collected powdery mildews their whole life. And they were really worried that nobody was going to continue their work. So they sent me all of the specimens they collected from throughout the world for their whole life. So most of these are from Germany and Japan. And these are what their collections look like. So the year they were collected, whether they're a type specimen. And my idea is to make the herbarium a global hub for powdery mildew research. So now I'm going to discuss, I always like to get fresh collections too. As DNA ages, it gets very fragmented. Oftentimes these specimens, DNA is fragmented into around 250 to 300 base pair pieces. So it makes it difficult to study. And that's why I always like to augment my collections with fresh specimens. And botanical gardens turned out to be, a really amazing place to collect fungal specimens. You have this great host diversity, so you're going to have this great fungal diversity. So between 2018 and 2022, I collaborated with horticulturists from 12 different botanical gardens throughout North America. Each color that you see here is a different powdery mildew species. I sequenced multiple regions for each one, so you can see there's a ton of diversity. At Harvard and at University of Washington is where I collected the majority of my specimens. That's where I did my PhD and my postdoc. But you'll notice I had collectors from all these other places as well, especially from Mexico. These are really important. The orange are undescribed species. So the majority of specimens collected from Mexico were undescribed. Just shows how understudied this group is. And people are always really excited to collect for you and to get, a lot of these are horticulturists, so they're excited to be on publications. So overall, I collected over 300 specimens. Of those, there were 130 different powdery mildew species, from 219 different host taxa, 30 undescribed species, and 45 first host pathogen records. So hosts that had never been reported to be infected by powdery mildew. But now I'm going to jump into 2 systems that I want to discuss. So this is powdery mildews on Quercus species, and this is really prevalent for here because we have so many different Quercus. It's a genus of over 400 different species, and it has centers of diversity in southern North America and Eastern Asia. I bet we have 50 different species just on campus here. And they're also, the powdery mildew is known to have serious ecological consequences. And there's been a lot of research about this, especially non-native powdery mildews that travel the world and infect non-native or hosts that they didn't co-evolve with. This is especially true in Europe, where there's an Asian powdery mildew attacking their native trees. And these trees will be completely covered white. And it's reducing photosynthesis, taking up nutrients, causing a lot of damage. So before I started, there were 6 powdery mildews that were described on oaks just based on morphology work. And just to give you guys an idea of this, there have been a lot of powdery mildews in the past 200 years described just based on morphology. In North America, there was around think 150, and before I started, there were only 20 sequences out of those 150. So we really don't know anything about the diversity or how this system works. And in this case, there were 6 powdery mildews on oaks described morphologically. But there was no reliable sequence data of any of the 6. So without sequence data, you're kind of playing blind. So in this study, I morphologically and phylogenetically analyzed collections from multiple herbaria, including the Larry F. Grand Herbarium, and they range from 1 to 140 years old. And this, I know you guys are mostly into genetics, but I think the morphology is also awesome, and I just want to point it out. This is a morphological feature that had never been seen before. These are these aerial hyphae. So hyphae are the vegetative state of the fungus. And usually they're like a mass of mycelium over the leaf, but for some reason on oaks, they make these hyphae that go straight up in the air, these aerial hyphae. And they had never been reported before. Okay, so then I went on to sequence 7 genes. Working with powdery mildews is really hard genetically because you're scraping the fungal material from the leaf. So you're picking up the entire microbiome of the leaf, all the other fungi, all the other bacteria. So we've developed, species-specific primers that most of the time only pick up powdery mildew. So we did 7 genes, and we built this phylogeny. So when looking at my phylogeny, this is a little blurry, but I'll walk you guys through it. The first name that you see here is the powdery mildew species. Then it's going to be on, and then on the host species. I list the host species and then the locality. So these are powdery mildews that were collected from, to me, this is pretty interesting. On the bottom, this is all on oaks, but from outside of North America. And on the top, we have all powdery mildews on oaks of North America. So they're all grouping together. That's not surprising. Of course, the specimens that are native to North America all evolved together. But what is really interesting is that in the past, like with most powdery mildews, they thought that these had a broad host range. But what we're finding is that these are all super host specific. So if you look here on the top, this is the white oak group. These are oaks that have round lobes. On the bottom is the red oak group, the oaks that have pointy lobes. So it's co-evolving with its oak hosts. And then you see a ton of host specificity. So we have this powdery mildew species that only occurs on one oak. This one also only occurs on Quercus garryana from the West Coast. And this just whole list. They're all extremely host specific. So it's telling this story of coevolution that I think is really interesting and that you can't really cross-inoculate this. Okay, so now I'm going to talk about a little bit more agriculturally important crop, and that's blueberries. And blueberries are really delicious, and people love to eat them. So this paper got a little bit of noise. I always say that, I want my pathogen to wipe out a really important economic crop so that I can be I can be a really famous plant pathologist. So one of the problems with fungi, especially plant pathogenic fungi, is that there isn't a great fossil record. So we have to try and piece together other hints of where these organisms are native to. And I'm just going to walk you through the steps that I usually go through. So I looked to see where that organism was described. So powdery mildew on Vaccinium was first reported in the eastern USA in 1832 as Ehrzyphe vaccinia. So that's one good evidence that this is native to eastern North America. It's reported on a range of Ericaceae hosts. And if you look here, the host range of the host that it infects tends to also be around the eastern North America. So this is another good. Its hosts and the powdery mildew were both first reported here in North America. And this is what's really interesting here, is that this powdery mildew had never been reported outside of America. But I would say 5 years ago, I got contacted by a big blueberry company. and they said, they saw one of my specimens on GenBank, one of my sequences. And they said, oh, I found something that matches with you from Peru and Morocco and China and Portugal. And it's all just within the past 10 years. Can you tell me how you identified this and what's going on? And I, of course, was super excited. Like, this pathogen that I study is spreading throughout the world, DOG blueberries. So I said, send me everything that you have, let's collaborate. And they did, and they were really helpful. And that's the data that I'll show you guys. But just to show you guys the importance of blueberries. This is how many hectares of high bush blueberry planting area was in 2021. And the company that I work with estimated that the amount of damage was between 200 and 2250 per hectare. And then you just multiply that out, and powdery mildew has the potential to cost the blueberry industry between $47 and $530 million per year. So this could be a pretty big issue. It's not going to cause the blueberries to not grow or go extinct. They're just going to have to spray a little bit more fungicide. And that's all what this calculation is. How much more fungicide are they applying? So now I'm going to go over the collection efforts. So we collected specimens from throughout the world. You can see these are everywhere. Here's where it had been unreported: South Africa, Peru, Morocco, Portugal, throughout China. These are top blueberry growing regions in the world. So it was from 13 different countries. And then we did the same process as with the Quercus powdery mildew. We sequenced 6 different regions. Some of these regions are linked to fungicide resistance. And this is the phylogeny that we built. And I'll walk you guys through this as well. So again, you're seeing the same pattern we saw in the Quercus. When you take a single loci, so like ITS is the general barcode for fungi. When you take the ITS marker, you're only going to see support right here. So you're going to see one big monophyletic group. And in the past, what they would say is that is one species and it attacks a bunch of different plants. Now we take this multi-locus approach, and we're finding that no, this is many species, because many separate host-specific species. On the top, this is just wild vaccinium species. And we found this all throughout the USA. Here, this is on Catalpa. These are big trees. We have them around here. This is powdery mildew on eucalyptus. And then we have here more on wild vaccinium. But the part I'm trying to point out is that we have 2 separate clades where we have powdery mildew on blueberries. So we have these 2 different forms of powdery mildew that are spreading throughout the world, and they're spreading to different localities. So this forma, which we're calling Forma vaccinii corymbosi, was found in Morocco, Peru, and Portugal. They were all found in the eastern USA. It's not surprising, we're seeing a bottleneck effect. And then here we have this one, which was found in China, Mexico, and the USA. So 2 different forms. And this is just opening research to look further into these 2 different. And I just want to give you guys an example of why this could be important. If you're a blueberry grower and you're trying to breed blueberries that are resistant to powdery mildew, you can't just take powdery mildew from this forma and expect that it'll be resistant to both of them. And even so, it might be if you're in Morocco, Peru, or Portugal, and you have one cultivar that's resistant to this forma and one that's resistant to this forma, you would send this one to those localities. So just another reason why the taxonomy is important. Just something else I want to point out, because I think this is so cool, is I do get a lot of photos of powdery mildew that they use as the cover photo, but usually it's like the beautiful Chasnodecia. But this was one of the first times they used the disease as the cover photo for the journal, which is beautiful in another way. But I was pretty pumped when I saw that. And then just back to the morphology a little bit. So this was another interesting finding from this paper, is that in Eastern North America, where this powdery mildew is native to, we always found this, the sexual stage, Ehr's-Iphy vaccinia. And with fungi, you need 2 mating types to make this, sometimes more, but for powdery mildew and for simplicity, you need 2 mating types. to make this sexual form. So basically you need these two strains to come together and then it forms this. But now, with all the powdery mildews that were introduced throughout the world, only one form was introduced. So it's only forming this asexual stage. And another question is, once we release this other form, how is this going to affect control? And how is it going to be spread? Is it going to be more virulent? Just other questions that really interest me. This caught my attention, and I had one of my workers look through herbaria to see one of the main powdery mildews is powdery mildew on wine grapes. It's heavily studied. And I was curious, there's been a lot of research that spread from Eastern North America to Europe, has been damaging the wine industry. So I was curious, how long was it from when the asexual stage was introduced till you start seeing the sexual stage? And he looked through herbaria and found that it was 60 years. So this was introduced in the 1860s, he found it, and everything until 1910 was the asexual form. And he looked at hundreds of powdery mildew specimens. But then around 1910, he started to see that switch. Yeah.
So in the herbarium, it can live on the specimen for 100 years?
It's not living, it's dried and dead.
It's what?
It's dried and dead.
And dead?
Yeah.
Okay.
So it's just like a dried plant.
Okay.
Yeah, but that was a good question. I get that a lot from USDA people about permits. That they're worried that it's going to spread, but this is dry, dead plant material. It's frozen in the freezer for 2 weeks. But yeah, so it took for powdery mildew on wine grapes, it took 60 years for the second strain to be introduced so it could form this sexual stage. And now I'm curious, how long is it going to be until this one? It'll happen, it's just when. Something else that was interesting is that we found powdery mildew, only the asexual stage, all throughout California. So again, this is telling me this is native to the Eastern North America, not the West Coast. And we're only seeing this asexual form. Something that I'm keeping an eye out on too is that powdery mildew on blueberries has never been reported from the Pacific Northwest. And so that for me, that's just a matter of time and we're going to start seeing that. So in conclusion, Yersinia vaccinii. This is actually work that was done by a professor here, Ignacio Carbone. He was really helpful with all this population genetics work that we did and that I didn't show you. But we showed that powdery mildew evolved over hundreds of thousands of years from a generalist. So it was able to infect all these different species to a specialist. I also didn't show this, but based on the single mutation in beta-tubulin in the site B region, we were able to see that it tends to be sensitive to FRAC1 fungicides and resistant to FRAC11 fungicides. And there are 2 different forms of Yersinia vaccinii spreading throughout the world from North America, impacting the blueberry industry. There's a lot of different directions you could take this. What we're really working on in my lab is to try and get full genomes of these. These are big genomes with a lot of repetitive elements, and they're unculturable, so it's very difficult, but I think it's going to tell a lot of really amazing stories when we get them. So we can evaluate the virulence of all these different powdery mildew forms infecting powdered blueberries, kind of how I alluded to before. Compare genomics of the different host-specific Yersinia vaccinii forms. These powdery mildews, if you take a single gene, they're 99.9% identical to each other, but they've evolved to infect different hosts and you can't cross-inoculate them. So that will be really interesting. Also, we can look at this same phenomenon in other systems, just like I showed you with oats. No one else is studying any of these powdery mildews, so it's really open. Here's just an example. We're seeing the exact same thing with strawberries. This is a big problem, especially in greenhouse-grown strawberries. And you can see that all the different, whether the powdery mildew infects different hosts, it's all forming these phylogenetic clades. So powdery mildew on cultivated strawberries is up here. Wild strawberries, roses, raspberries, thimbleberries, blackberries, they're all genetically separate. So we could do this same approach on a different system. So here's just kind of what I alluded to, what something that interests me is that you can look in a single region, the beta-tubulin region, and a single point mutation can correlate with fungicide resistance. And to me, that's really interesting, and it opens up a lot of research that can be done. I did this during the pandemic. I couldn't go in the lab. I was working at the USDA, and we were just brainstorming, like, what are we going to do? How are we going to do this research? And something that is ingrained in me, from Herbarium Specimen, is trying to take this time approach. So how are populations changing over time? And so that's kind of what I wanted to do here. How is FRAC1 resistance changing over time? And how can we do this genetically? Look at this genetically. So I just mined GenBank. So I obtained 2,647 specimens from GenBank and the peer-reviewed literature. I chose the FRAC1 fungicide resistance because TUB, the loci that's related to this, that has that point mutation, is often used in taxonomy studies. So people are sequencing it a lot. So most of these specimens were not sequenced for fungicide resistance studies. They're just randomly sequenced. And I looked at, in total, I looked at 12 genera of fungal phytopathogens in 53 countries, collected on over 200 hosts between 1926 and 2020. And this is the data that we found. So this is the percent of the pathogens that had this point mutation. So you can see, of course, prior to 1980, FRAC1 fungicides really weren't used very often, and we're not seeing any resistance in the populations. Then as we start using more fungicides, we're starting to see a lot more resistance. But then it dipped here. And to me, this was showing that people are not stopped using this type of fungicide, and that the populations are rebounding. So that would mean that this mutation or fungicide resistance has a fitness cost for the pathogen. So I sent this into a review, and reviewer 2 wanted more evidence of what I was saying. And at first I was really upset, because they denied my paper. But then I thought, I actually think that was a really good critique. I think that we need more evidence to show that, something happened here, like that we stopped using these fungicides. So I went to all the publicly available databases for fungicide usage. So there's 2 states that have this, and that's California and Arizona. So they mandate growers to report the fungicide that they use. So here, I got the data, and you can see in California and Arizona, we're seeing this same dip. And I thought that is amazing. Exactly what I hypothesized. And what's interesting is that dip is happening 10 years before we're starting to see the population, fungicide-resistant population decline here. Since then, I actually just talked about this to an IPM group around 6 months ago, and someone told me that they just did research of this in vivo and found they were able to show the fitness cost. So I think that this was really a nice full circle for this paper, and it's one of those times where the reviewers really helps you with your research. And then last, I just want to discuss using herbaria and fresh specimens to understand extinction events. And I just think this is such a cool story that I'm trying to unravel right now. So this is Phylactinia, a genus of powdery mildew that GenBank told me was alien DNA. And there has been a species of this that affects the American chestnut. And I don't know if you guys have heard the story of the American chestnut, but there's another fungal organism, Cryphonectria parasitica, which has decimated this tree. It's still around, but once it gets to a certain height, it dies back, and you only see little shrubs on the ground. This powdery mildew Phylactinia, I often see it on more mature plants. Cryptonectria parasitica was introduced in the early 1900s. This powdery mildew was described off material of the American chestnut in the early 1800s. And the last time it was collected in herbaria was around 1870. So between 1800 and 1870, we're seeing a ton of collections of this. I see it all throughout herbaria. But then all of a sudden, it stopped. There's been no collections of it in 150 years. And I have multiple people. Andrew here is looking through the plant herbarium here to try and see if we can find it. I have a colleague in Michigan State going through one of the last natural forests in Michigan, natural stands of this plant, to try and find this powdery mildew. So I'm bringing together all this evidence to try and show that this powdery mildew might be extinct. And this might be one of the first reported cases of one fungal pathogen causing the extinction of another fungal pathogen. So I think that this will be a real good paper with a lot of clickbait if we can really prove that this is extinct. But I'm trying to pull out all the resources we can before we make that conclusion. But basically, the question is, could one plant pathogen have caused the extinction of another plant pathogen? And my feeling is yes. And with that, I'm happy to take on questions. Here's my lab. They've all been great help with all this research. And thank you.
We have time for questions. What makes powdery mildew a specialist? Is it abortion of the spore germination? Is it lack of some sort of recognition?
I think it's lack of some sort of recognition. There really hasn't been that much study on it. I think there's just so much crosstalk between the host and the pathogen. It will germinate. You can get powdery mildew to germinate on a slide, but it won't form in a pressoria, which is like this peg that goes into the leaf. And I think that the powdery mildew is putting out a signal, and then the host puts out a responding signal, and the powdery mildew does a responding signal, and it goes back and forth. And this is why you can't culture it, because even when people have these mixtures of plants and in the media, it still doesn't work. So there's something going on where they're instigating these different effectors and molecules to be secreted.
And how does it overwinter?
In that fruiting body, the Chasmothecia. So it's a protective structure that allows it to overwinter.
So it's in soil.
No, it's on the leaf. So that's a good question. So it can do multiple ways. So on woody plants, it can grab onto the buds. So then when the new leaves emerge in the spring, it just connects onto it. But also on herbaceous plants, the plants die down, the powdery mildew is on the ground in the soil, and then when the plants come up the following year, it hooks onto the plants. So this is a big group. I always like to mention this. When we're talking about one powdery mildew genus compared to another, you could be talking about like a frog and us. So there are a lot of genetic differences between these organisms, even though they look very similar to us. But yeah, good questions.
Is there anything that can survive on, like saprotrophically on dead stuff, or?
No, nothing. Nothing in this group.
Yeah. How far do you have to get out?
So that's a really interesting question. Like, what is the sister group of powdery mildew? We really don't know. But I wish I could show you guys. The current estimate, looking genetically, is that it is a saprobe. That it evolved from a saprobe, but it's a cup fungus. So like it looks like a little mushroom. But morphologically, it just doesn't make sense. I think we're missing all these links. But hopefully I'll figure that out one day.
Are there cost-effective ways that governments should institute or that nurseries should be following to prevent powdery mildew strains from being introduced when the plant germplasm is moved around the world?
Yeah, I think that that's a good question. This is what I always say to that is, these companies are growing their nursery stock in one country and then sending it throughout the world. Of course, you're going to be bringing the pathogen with it, even if it's all cleaned up. Powdery mildew mycelium can also overwinter within the buds of the plant. So you think you have this clean plant with no leaves or anything, it opens up in the spring and it's infected. So I think maybe growing nursery plant material in your final destination would be better than growing it all in one place and sending it everywhere. Just one of my thoughts. prevent the spread.
Is there any evidence of hybridization between related species?
Okay, that's another thing I really want to answer. So that has never been shown before in powdery mildews. It's, but I think it must be happening.
So
And-
co-infections can happen, right? Because for the cross to occur, they have to be in the vicinity of one another.
Yeah, exactly, yeah. But like between species, we haven't seen any hybridization happening. But that could be really interesting if we could show that. I think we did that for that year's IT vaccinii paper and said that there wasn't any hybridization.
I liked your use of arboretums as a source of botanical gardens as a source of fungal diversity, but it seems like right now it's just limited to the continent of North America. There were some of those other South American, African, Middle Eastern continents. Do you have any plans to use botanical gardens on those continents to maybe pull some fungal diversity?
Yes, absolutely. So the thing is that this is a NSF funded grant to look at the diversity in North America.
Gotcha.
So the next step is to do the worldwide approach. You know, if NSF is still accepting it.
So on the last point, the extinction, I mean, American chestnuts, were almost completely obliterated in our view. So you'd expect anything that grows on American chestnuts to be almost completely obliterated too. And, how do you tell the difference between almost completely obliterated and completely obliterated? I mean, extinct and or very, very, very rare. There's no, you can't, how can you prove something's extinct?
Yeah, I think that that's a great point. I can't really prove that it's extinct, but I can just do the best effort possible to search for it, look through herbaria, look through fresh specimens, and then if I find it, if I can't find it, say there's a good chance it's not around anymore. Yeah, but there's no way for me to 100% conclusively say that this pathogen is gone. Or what if this one had a little bit of a broader host range? Sometimes we see that and it jumped to something else and we'd never be able to make that connection. Unless I sequence the herbarium specimens from the ones from the 1850s. Any other questions?
All right, well, let's thank Dr. Bradshaw.